I'm going to talk to you today, about drug R&D. I think it's going to be a little bit of a different kind of talk for ICML. Okay, so I work in Genentech. We're a biotech company, and we focus on discovering and developing transformative medicines for patients like Lysandra and Lynn and Gary, who live with diseases like breast cancer or lung cancer or multiple sclerosis. And in each of these, we aim to tackle foundational causal pathways and targets, and to discover and deliver therapeutics that can change patients' lives. So, for example, think about something like inavolisib. That's a drug. It targets PI3 kinase, which is one of the most commonly mutated cancer-causing proteins. And in this way, it reduces the growth, the survival, and the proliferation of malignant tumor cells. So there's been many attempts to target PI3 kinase alpha, but in the lab, inavolisib has a higher potency and selectivity. And in a phase 3 trial that combined inavolisib with palbociclib and fulvestrant in the first line, this translated into transformative outcomes for patients with HR-positive, HER2-negative breast cancer with mutant PI3 kinase. Now, I'll tell you about another drug called duradestrant. It's an oral potent and selective degrader of the estrogen receptor, also called the SERD. It binds and immobilizes the estrogen receptor, and it inhibits ER signaling. Again, in the lab, duradestrant has the best coverage of the target, and it allows us the lowest possible human dose. In the clinic, it was well tolerated, and in the lidERA phase 3 trial that read out late last year, it reduced invasive recurrence and death by 30% compared to the standard of care. Now I'll move to lung cancer. Divarasib, it targets another key oncogene called KRAS with a G12C mutation. It locks it in an inactive state. In the lab, it's 5 to 25-fold more potent. It's 10 to 50-fold more selective than other inhibitors. In a phase 1 trial in the clinic, it showed strong single-agent activity. And just over a week ago, we reported the results of the phase 3 trial, Crescendo 1, that showed that divarasib helped people with advanced metastatic non-small cell lung cancer with this kind of mutation live longer without disease worsening and live longer overall compared to other approved G12C inhibitors. And now I'm going to move from cancer to multiple sclerosis. It's an autoimmune disease. Fenebrutinib selectively inhibits a gene called BTK to suppress immune signaling in 2 different cell types that are important for multiple sclerosis, microglia and B cells. In patients, fenebrutinib is also the only BTK inhibitor that achieves near maximal inhibition of these cells inside the central nervous system. And in phase 3 trials, it significantly reduced the relapses and the brain lesions relative to teriflunomide. And it translates from MS patients to having an average 1 relapse approximately every 16 to 19 years. That's what we mean by transformative medicines that we discovered and developed in order to help patients like Lynn and Gary and Lysandra. And we do this and work to discover them across 5 different major therapeutic areas. But what I want you to take from this is that doing that remains really hard. This is not just the case in Genentech, and it's not just about one particular single step in making medicines. Everything is really hard. All the steps have to align, the right target and medicine and dose and molecule and patients. And all of these things are not just time-consuming. Most of the time, they actually don't succeed. And that begs the question, why? Okay, so it's because biology and medicine are actually really hard in 4 specific ways. The numbers are big. The system spans multiple scales. We can measure it in many different ways, but not at once. And it's super varied. It's never quite the same. And I'll take you through these. So first of all, when I say the numbers are big, I mean they're really big. There's about 37 trillion cells in the human body, 100,000 disease-associated loci that were mapped to date, about 10 to the power of 19 ways in which they could hypothetically combine in 5-way combinations. And when you start thinking about medicines, which are mostly molecules, the numbers get really big. there's about 10 to the 60 possible drug-like small molecules, maybe 20 to the power of 32 relevant therapeutic-like antibody sequences that we could consider, billions of people and thousands of diseases. And we can never, ever test all of these possibilities in a lab or an entire human population. It's so not surprising then that it's hard to find targets and make drugs. Okay, but that's just one problem. Now it's also multi-scale. We have thousands of cell types in intricate tissue structures. They're governed by all of these genes. That's a different level of organization. And they have all of these variants that increase the risk of disease by affecting proteins that can take many different forms and be in many different places across many different disease phenotypes. And now to make it even worse, we don't have just one set of unifying measurements. Even within one level of biological organization, like the cell, there's many, many ways to measure a cell, and I can't do that all at once, many views. And then, finally, even the problems that are kind of similar and we kind of learned how to solve, they're never exactly the same, they're varied. So even if we did something multiple times, it's hard to automate it, not in the lab and not in analysis. So all of that makes the toiling just hard to predict, hard to interpret, hard to generate, and then the humans, us, have to somehow wade through all of this. Okay, so now compare that, big numbers, multi-scale, multimodals, and so varied, to the 4 success stories I described to you in the beginning. And they look so tiny, not anything close to these kinds of numbers. So that's why it's so hard to make transformative medicines. But fortunately, all of these things actually have properties that make AI very relevant. Yes, the space is nominally huge, but it's structured and its real dimensionality is a lot smaller. Well, bingo, AI is good for that. Yes, it's multi-scale and all the transformations are non-linear. AI is good for that too. Yes, it's multimodal and I have a lot of views. Well, AI is pretty good at that. And yes, it's varied and I can't exactly automate it, but agentic AI is starting to look pretty good on that as well. So could AI help take something that feels nearly intractable and often fails and somehow make it better? Now, like any hard problem, people have a lot of ideas what's most important. So some people say it's all in the models. Of course, some people say it's all in the data. Some people say it's all in the models. And some people, of course, say it's all about the experiments and the synthesis and the human biology. Well, they're all kind of right and all kind of wrong. Each of these pieces is necessary, but none of them alone is sufficient. What we need to do is to put these together in what we call the lab, or sometimes the clinic, in the loop. Start with an experiment, collect data, train a model, use the model to define the next set of experiments, and repeat. And as we do that, we can iterate our way to drug discovery and development. So this is what we've been building over the almost past 6 years in Genentech.
更多演讲实录与文章已收录至雷锋网 ICML 2026深度专区，现已全面上线。专区不仅全面收录了重磅论文的工程化解读、专家前沿演讲，更将持续更新前方记者的第一手会议动态。扫描下方2维码或进入雷锋网 ICML 专区即可查看。